Good morning, students. Today we start the new chapter, that is respiration in plants. Respiration. Respiration is the stepwise oxidation of complex organic molecules and release of energy as ATP for various cellular metabolic activities. Compounds oxidized are respiratory substrates which are carbohydrate, proteins, fats and organic acids. Actively respiring regions are growing regions like floral and vegetative parts, germinating seeds, stem and root apices. Types of respiration. There are two types of respiration, external respiration and cellular respiration. Cellular respiration is of two types, anaerobic and aerobic. Anaerobic means in absence of oxygen, aerobic means in presence of oxygen. External respiration. The respiration involves exchange of gases between the organism and external environment. The plants obtain oxygen from their environment and return carbon dioxide and water vapor into it. This mere exchange of gases is known as external respiration. External respiration. In plants, the atmospheric air moves in and out by simple diffusion that takes place through the general body surface of the plants, means stems, roots, fruits and seeds. Lenticels, opening present on the bark of tree trunk, and stomata present in the leaves and young stem. So, from this very parts, the gaseous exchange takes place in the plants, and that is external respiration. External respiration. In plants, the atmospheric air moves in and out by simple diffusion that takes place through the various structure as shown in the picture. Lenticels present on the tree trunk. Cellular respiration. The biochemical processes which occurs within cells and tissues and oxidizes food to obtain energy is known as cellular respiration. Various enzymes, biocatalysts, catalyze this process. On the basis of utilization of oxygen, cellular respiration is of two types, anaerobic respiration and aerobic respiration. Steps of cellular respiration. See the first step, that is the glycolysis, where sugar is broken down into two molecules of pyruvic acid. That pyruvic acid undergoes for anaerobic respiration and forms ethanol and lactic acid, or in the case of aerobic respiration, it goes to Krebs cycle. Now see the glycolysis or EMP pathway. Whether or not oxygen is available in the cells, the breakdown of glucose is initially always anaerobic. It is common to both aerobic and anaerobic respirations. It involves oxidizing glucose to two molecules of pyruvic acid through a series of enzymatically controlled reactions occurring in the cytosol. Initial substrate is glucose, either from photosynthesis as in the plants or from carbohydrate digestion as in animals. See the steps of glycolysis. Glucose, that is a 6 carbon compound, first of all changes into glucose 6 phosphate in presence of one molecule of ATP. Then very soon glucose 6 phosphate again changes into fructose 1,6 diphosphate by consuming 1 ATP more. Then fructose 1,6 diphosphate broken to two molecules of 3 carbon compound, and that finally changes into pyruvic acid by releasing hydrogen. The balance equation is glucose plus 4 ADP plus 4 inorganic phosphate plus 2 NAD, that is a coenzyme, 2 pyruvic acid, 4 ATP and 2 NADH is formed. 2 molecules of ATP were used up in the initial stage of glycolysis, thus the net gain of ATP during glycolysis is 4 minus 2, that is 2 ATP. Also 2 NADH plus H plus are produced. Thus, we see that only a small amount of energy is released at the end of glycolysis. Fermentation under anaerobic conditions or insufficient supply of oxygen, microbes and plants carry out fermentation. Fermentation involves reduction of pyruvic acid to ethyl alcohol and carbon dioxide as in yeast, or to lactic acid as in muscle cells of animals, and oxidation of NADH to NAD+. Pathways of anaerobic respiration. Glycolysis, that is in the case of aerobic respiration, 2 pyruvic acid is formed. Lactate formation, that is also anaerobic respiration in the muscles, that also changes into 2 pyruvic acid. Then alcoholic fermentation, as in the case of yeast, there also 2 pyruvic acid is formed. Then in the case of muscle cells, pyruvic acid releases lactic acid, and in the case of yeast, that pyruvic acid finally forms 2 molecules of ethanol. So, net result in the case of glycolysis, that is 1 molecule of pyruvic acid, 2 ATP, 2 NADH. In the case of lactic acid fermentation, glucose changes into lactic acid and 2 ATP, and in the yeast 2 ethanol plus 2 ATP. Fate of pyruvic acid in aerobic respiration. In presence of oxygen, pyruvic acid enters the mitochondria and is decarboxylated, that is removal of carbon dioxide, and dehydrogenated, a removal of hydrogen, to acetyl coenzyme A. Acetyl coenzyme A is thus the connecting link between the glycolysis and the next series of reactions that yield more energy in the form of ATP. Acetyl coenzyme A can also be generated from fat and proteins. Krebs citric acid cycle. Acetyl coenzyme A is the molecule entering the Krebs cycle, taking place in the mixing of mitochondria, matrix of mitochondria. Details of the cycle were worked out by Sir Hans Krebs in the year 1930. It is also known as tricarboxylic acid cycle or TCA cycle. See the various steps of Krebs cycle. Pyruvic acid changes into acetyl coenzyme A, then acetyl coenzyme A combines with oxaloacetic acid to form 6 carbon atom molecule, that is citric acid. Citric acid very soon changes into alpha glutaric acid, and a series of reaction finally produces oxaloacetic acid. In this Krebs cycle, 2 pyruvic acid plus 8 NAD plus 2 FAD plus 2 ADP consumed and releases 6 CO2 plus 8 NADH plus 2 FADH2 and 2 ATP. Respiratory chain or electron transport chain. The hydrogen carriers now move to the inner membrane of the mitochondrion. This membrane has the folds called cristae, which increases its surface area. Hydrogen carried to the cristae undergoes stepwise oxidation using molecular oxygen, and energy is released. In a series of small steps, some of this energy is used to make ATP from ADP and inorganic phosphate. This is called oxidative phosphorylation. During these reactions, the hydrogen is split into H plus and electrons, which are accepted by a series of hydrogen or electron carriers ending with oxygen. This series of carriers constitute the respiratory chain. See the respiratory chain or the electron transport system. Hydrogen or electrons at higher energy level are passed from one carrier to the next, moving downhill in energy terms until they reach oxygen, the final acceptor of electron, which as a result of reduced to water. At each transfer, some energy is released, and in some of the transfer, this is used for the formation of ATP. The final steps involve cytochrome oxidase enzyme, which hands over the electrons to the proton before being accepted by oxygen to form water. Summary of aerobic respiration. How the 38 ATP molecule is produced in the respiration. During glycolysis, 2 NADH2 is produced, 6 NADH2 and 2 FADH2 in the Krebs cycle, and 2 NADH2 from formation of pyruvic acid to acetyl coenzyme A. This undergoes in the electron transfer system, and 6 CO2 is consumed and forms 34 ATP, 2 FAD